Biochemistry. Carbohydrate metabolism is a chemical process that involves the processing of carbohydrates in the body to produce energy, form body structures, or be stored for future use. Carbohydrates are one of the main sources of energy for the body, and the process of carbohydrate metabolism occurs in various complex stages in cells. This process involves a series of biochemical reactions related to the breakdown, formation, and storage of carbohydrates. Here is a detailed explanation of carbohydrate metabolism. 1. Carbohydrate digestion and absorption. Carbohydrates in food, such as starch and disaccharides, e.g., sucrose, are digested into monosaccharides, glucose, fructose, and galactose, that can be absorbed by the body. The process of carbohydrate digestion begins in the mouth with the help of the enzyme amylase, which breaks down starch into maltose, a disaccharide. In the small intestine, other enzymes, such as maltase, sucrose, and lactase, convert disaccharides into monosaccharides. The monosaccharides formed, especially glucose, are then absorbed into the blood and carried to the body's cells to be used as an energy source. 2. Glycolysis. After glucose is absorbed and enters the cell, it undergoes a series of reactions known as glycolysis, which occurs in the cell's cytoplasm. Glycolysis is the process of breaking down glucose, C6 oxide, into two molecules of pyruvate, C3H4O3, producing a certain amount of energy used by the cell. The glycolysis process takes place in 10 steps, which are grouped into two phases. A energy investment phase, namely, in this phase, 2 ATP molecules are used to phosphorylate glucose, converting it into glucose 6-phosphate and then into fructose 1,6-bisphosphate. B. Energy formation phase, namely, fructose 1,6-bisphosphate is broken down into two 3-carbon molecules, namely dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. G3P is then converted into pyruvate, producing 4 ATP molecules, 2 ATP molecules for each G3P molecule, and 2 NADH molecules, electron carriers. Overall, glycolysis produces 2 ATP, 2 NADH, and 2 pyruvates from each glucose molecule. 3. Formation of pyruvic acid in the citric acid cycle. After glycolysis, the pyruvate produced enters the mitochondria for further processing. A. Decarboxylation of pyruvate, namely, pyruvate is converted into acetyl-CoA by the enzyme pyruvate dehydrogenase. During this process, pyruvate loses one carbon atom in the form of carbon dioxide, CO2, and produces NADH. Acetyl-CoA then enters the citric acid cycle, Krebs cycle or TCA cycle. B. Citric acid cycle, Krebs cycle. Namely, acetyl-CoA combines with oxaloacetate to form citrate, citric acid. In a series of reactions involving oxidation, decarboxylation, and electron transfer, citrate is converted back to oxaloacetate, producing 3-NADH, 1-FADH2, 1-ATP, in the form of GTP, and 2-Colorado-2 molecules as byproducts. This cycle takes place in the mitochondrial matrix. 4. Electron transport chain and oxidative phosphorylation. The products of the citric acid cycle, namely NADH and FADH2, carry the electrons needed to produce ATP through the electron transport chain. This chain occurs in the inner membrane of the mitochondria, the inner membrane of the mitochondria. A. Electron transport, namely, NADH and FADH2 donate electrons to the electron transport chain, which consists of a protein complex in the mitochondrial membrane. Electrons move through this chain, moving protons, H+, into the intermembrane space, creating a proton gradient. B. Oxidative phosphorylation, namely, the proton gradient formed is used by ATP synthase to convert ADP to ATP in a process called oxidative phosphorylation. The electrons are finally accepted by oxygen, which binds the protons to form water. As a result of this process, each glucose molecule can produce about 30 to 32 ATP molecules. 5. Gluconeogenesis. Gluconeogenesis is the process of glucose synthesis from non-carbohydrate precursors, such as lactate, pyruvate, amino acids, and glycerol. This process occurs mainly in the liver, and to a lesser extent in the kidneys, and serves to maintain stable blood glucose levels, especially when the body is fasting or under stress. Gluconeogenesis is the reverse of glycolysis, although there are some different steps because glycolysis has reactions that cannot be reversed. 6. Glycogenolysis and glycogenesis. Glycogen is the storage form of glucose in the body, which is mainly stored in the liver and muscles. The two main processes that regulate this glucose reserve are a glycogenolysis, namely, the process of breaking down glycogen into glucose 6-phosphate, which can then be used in glycolysis or converted into free glucose in the blood, especially in the liver. Glycogenolysis is triggered by hormones such as glucagon and epinephrine. B. Glycogenesis, namely, the process of synthesizing glycogen from glucose 6-phosphate. Glycogenesis occurs when the body is eating or after eating, and excess glucose is stored in the form of glycogen in the liver and muscles. This process is influenced by the hormone insulin. 7. Fructose and galactose metabolism. In addition to glucose, the The body also metabolizes fructose and galactose, although their metabolic pathways are slightly different. A fructose, namely, when in the liver, fructose is converted to fructose 1-phosphate, which is then broken down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which can enter glycolysis. B. Galactose. Namely, galactose is converted to galactose 1-phosphate, which then interacts with glucose 1-phosphate to form glucose 6-phosphate, which can be used in glycolysis or other processes. 8. Regulation of carbohydrate metabolism. The process of carbohydrate metabolism is tightly regulated by various hormones, including insulin, namely, produced by the pancreas. Insulin lowers blood glucose levels by increasing glycolysis, glycogenesis, and glucose uptake by cells. B. Glucagon, namely, also produced by the pancreas. Glucagon increases blood glucose levels by stimulating glycogenolysis and gluconeogenesis. C. Epinephrine, adrenaline, namely, triggers glycogenolysis to provide quick energy in stressful situations. Conclusion. Carbohydrate metabolism involves a series of complex processes to obtain energy from carbohydrates. Starting from digestion and absorption of carbohydrates, glycolysis, the citric acid cycle, the electron transport chain, to the storage and synthesis of glucose. All of these processes work together to ensure that the body gets enough energy to carry out various biological functions. Proper hormone regulation is essential to maintain the balance of carbohydrate metabolism and ensure the availability of energy needed by the body.